Hello, everybody. I have a short disclaimer before we start today's class. There is some construction going on nearby, so there might be slight disturbances like that one. But I assure you that that is not going to be the majority of today's lecture. Okay, so I'll take care of that whenever I'm speaking so that there's no overlap. Okay, having said that, I'll just share my screen and then we can admit more of your fellow learners who are joining. And yeah, first let me share my screen. Okay, I hope the slideshow is visible to everybody who's attending, right? I'll just remove this view. I think this is okay. Yeah, currently I'm sharing my screen for people who are viewing my lecture for the first time or the learners who are joining us in the sixth week of this particular course, that is cell biology, cellular organization, division, and processes. Let me give a brief introduction about myself. I am Arveen Kaur. I am a PhD scholar at Department of Biological Sciences and Bioengineering at IIT Kanpur. Along with this, I am also a Prime Minister's Research Fellow, for which I have been assigned, as a part of which I have been assigned as a tutoring assistant or teaching assistant by NPTEL for this particular course that is being conducted by Professor Shikha Laloria. So I am sure you have been going through the lectures that ma'am has been uploading, right? And they are very, very informative lectures. What we do in these practice sessions are is that we will go through the previous year's questions pertaining to whatever week we are in, right? So currently, week six lectures had been released, and now you have the assessment that is open for you guys to solve and submit for week six, right? So since that is an open assessment or an open assignment, and the final date has not been surpassed yet, so we are forbidden to actually discuss questions particularly from those assignments. But what we can do is that we have access to the previous year's assignments, right? So last year for this week six lectures, there were some questions that might have been given, and we can basically go back and we can solve those questions. It might be based on the similar concept on which the questions have been asked this week as well, right? So that might help you, and it might also help you if you have registered for the exam for this particular course, right? So it is just like practicing a few additional questions. So whenever we'll be solving these questions, what we'll do is if there is any concept that is linked to it, we will go back and we will also study that concept again just for a revision sake, right? So that is going to be the first part of today's lecture. In the second part, what we'll do is that we will discuss a few extra questions and a few extra concepts apart from what have already been taught to you in Professor Laloria's lectures, right? So we are going to discuss those as well. So, yeah, let us begin with the first part. So, the first question from the previous year's assignment for week six is, each nucleosome core particle is separated from the next by which one of these? So, is it the histone tail or the linker DNA or the linker RNA or the SSB proteins? So I'm sure you guys have gone through the lectures that were delivered by ma'am and a few other professors, right? So this is a pretty straightforward question. I hope you know the answer. We'll just jump onto it. So the answer for this one is linker DNA, right? So what is a nucleosome core particle? A nucleosome core particle basically comprises of histone proteins and DNA that is wound around these histone proteins, right? And these units are separated by free DNA, as in DNA that is not bound by histones, but may be bound by some other non-histone proteins, right? And this DNA is known as linker DNA. So this is a representation that I used in week 4 lectures as well, right? So over here, this represents the beads on string structure in a schematic or illustrative way, right? Over here, these yellow, circles are the histone proteins. Histones are octameric complexes, as you all know, right? The other colored structures over here are the non-histone proteins, and you can see that there are different kinds of non-histone proteins that are actually present, right? And you can see in between these, right? There are histone proteins are also bound by DNA. That is, DNA is wrapped on them, right? Apart from this DNA that is wrapped on these histone proteins, there is also some free stretches of DNA which might be interacting with non-histone proteins or might not be interacting with them, right? These are the free DNA or the linker DNA. Linker DNA is basically connecting two nucleosome core particles, right? Or separating two nucleosome core particles. So whatever terminology you want to use is correct, right? But the answer for this one is linker DNA. Yeah. So the second one is cleavage of MCD1 by separase triggers which one of these? So the movement of sister chromatids into opposite halves of the cell during anaphase. B, the movement of sister chromatids into opposite halves of the cell during metaphase. C, the movement of both sister chromatids into a single half on the cell during anaphase. Or D, the movement of both sister chromatids into a single half of the cell during metaphase. So for this, you will have to remember, or you will have to recall what is separase, right? What does separase do? What is MCD1? Have you ever come across this particular word, right? So it is pretty apparent it might be another protein, right? So what is this protein involved in? So think about that and you might stumble upon the correct answer, right? If you don't already know it. Okay, so let us move to the answer for this particular question. So the answer is the cleavage of MCD1 by separase triggers the movement of sister chromatids into opposite halves of the cell during anaphase, right? So I will explain what MCD1 is. Before that, let us see that even if you did not really know this answer, would you have narrowed it down to the correct answer, right? So what is the second option that is given over here? The second option says the movement of sister chromatids into opposite halves of cell during metaphase. So you know that in metaphase, this movement of sister chromatids into opposite poles does not occur, right? That is a characteristic feature of anaphase. At metaphase, the sister chromatids will be lined up at the equator or the equatorial plate, right? So this is the incorrect answer, right? If you know the basics of cell cycle and mitosis, you will know that this answer cannot be correct, right? And then C is the movement of both sister chromatids into a single half of the cell during anaphase. Now, generally in a normal cell, this will not occur, but we can still consider this for now, right? Why? Because maybe MCD1 is normally not separated by separase, right? It might have, but over here, since no kind of mutation is also talked about, you can actually eliminate this one also, right? The final one again says movement of both sister chromatids into a single half of the cell during metaphase. Again, during metaphase, the movement of sister chromatids to opposite poles will not occur, or even to one pole will not occur, right? So this one is also automatically omitted. So even if you don't exactly know what MCD1 is, you can come down or narrow down your answer to A or maybe A and C, and then you can choose one, right? But for now, uh, you might be aware, if you recall correctly from your lectures that you have attended by Professor Shikhala Laura, right? That MCD1 is one of the non-histone proteins, right? So there are multiple non-histone proteins. SMC protein complexes are one of them. SCC proteins are one of them. RAD21, MCD1. These are all non-histone proteins, right? And these proteins, the ones that I have just named, are the non-histone proteins that are this MCD1, SCC proteins, and RAD21 are the non-SMC non-histone proteins which are associated with cohesin. Right, so whenever you might have studied about cohesin before, either of these you might have encountered, right? SCC13, RAD21, or MCD1, right? These are all non-SMC, non-histone proteins that are associated with this cohesin complex, right? And now, since they are associated with this cohesin complex, what does separase do? Separase basically cleaves or opens up the cohesin complex such that the sister chromatids can now move to opposite poles, right? So, what like if you know that it is a part of cohesin, you know that separase, the action of separase will basically result in movement of sister chromatids into opposite halves of the cell during the anaphase, right? So this one is thus the correct answer. Okay, so moving forward. Yeah. So this was another illustration I have kept from this particular paper, wherein ESP1 is a kind of separase, right? So ESP1, when it is bound to this particular subunit, you don't have to dwell upon its name. So, or learn its name for that purpose, right? When it is bound to a certain subunit, it is like non-functional. But when the APC complex or APC related CDCs and CDKs are activated, what happens? It basically becomes okay, sorry. It comes into an active phase, right? In this active phase, it will. So this could be a kind of securant. You remember we have studied this separate securant complex in earlier practice sessions, right? So whenever the APC cyclins and CDKs are activated, it will go and it will ubiquitinate the securant, which will basically render this particular separase ESP1 free, right? Once it is rendered free, it will cause cleavage of MCD1 and this will lead to anaphase onset, right? So that is it. Coming to question number 3. So question number 3 says the condensation of chromosomes during mitosis is essential because 1, it prevents the entanglement of chromosomes with each other and the cleavage of DNA during cytokinesis. 2, it enhances the efficiency of replication. 3, it prevents cells from entering meiosis. Or D, none of the above. So go through the question very nicely and we'll discuss the answer also. Okay. So the answer for this particular question is the first option, that is the condensation of chromosomes during mitosis is essential because it prevents the entanglement of the chromosomes with each other and the cleavage of DNA during cytokinesis, right? So again, this is something that you should know, but the other options, let us go through them, that can they be true, right? So it says that condensation of chromosomes enhances efficiency of replication. Now, can that be true, right? First of all, during mitosis, replication doesn't occur, right? Replication occurs in S phase, right? When this DNA is in the S phase and it has to be replicated, is it in the chromosome state? No, it is in the chromatin state, right? So the it needs to be in a decondensed and more accessible form for replication to actually take place, or replication machinery to be able to come and bind and replicate, right? Condensation will definitely not enhance the efficiency of replication, right? Also, it will not prevent the cells from entering meiosis, right? That decision also takes place very early on in the G1 phase itself, right? In the G1 to S phase transition, this particular decision takes place. The condensation of chromosomes has nothing to do with it. Even during meiosis, the chromosomes will be in a condensed state, right? And none of the above is not the true answer, because the first statement that is given over here, that if the chromosomes are in a very condensed form, so they will be able to be packaged very nicely, and it will also help because the chromosomes have to move, right? First they'll be spread out across the cell, then they have to come to the equator or the central plate, then they have to move to the opposite poles, right? So much of movement has to happen. So during this movement, we don't want these chromatin threads to become entangled within one another, right? So that is why this is the correct state. Okay. Uh, for representation purpose, I have put this particular illustration that is taken from Albert's, the molecular biology of the cell. Sorry. So over here, you can see that, for example, the DNA to begin with, if we talk about a DNA double helix, it will hardly be 2 nanometer in diameter, right? But when we talk about the chromosome, it will be around 1400 nanometer in diameter. But on the contrary, this is a short region of the DNA double helix, right? It is a very, very long double helix. And in some cases, the longest DNA, for example, in humans will be around 10 centimeter long, right? So that 10 centimeter long DNA has to fit inside a nucleus, right? Which is microscopic in size. So for that, there is some kind of packaging that occurs. When it packages into the histones in a beads on string form on the chromatin, what happens is it becomes around 10 to 11 nanometer in diameter, right? It is further packaged into this 30 nanometer structure that we will discuss about a lot in today's second part, right? And this is further condensed into or folded into loops. Like this structure is further folded into loops, and these loops are further, even compressed further, and they finally form a entire mitotic chromosome, right? So you can see that uh, this big of DNA formed only this much of this Bison string, while this big of Bison string formed only this part in this particular structure, and that just represented this big part in this chromatin fiber, and this looped chromatin fiber further represents only this bigger part in the entire chromosome. So you can just extrapolate how big this particular DNA will be, that it will, encompass the entire length of this chromosome, right? So what is the net result out of this? Each DNA molecule has been packaged into a mitotic chromosome that is 10,000 fold shorter than its fully extended length. Right, so that is a huge range of folding that is seen in the mitotic chromosomes, right? Moving on to question number 4. The question is, select the correct statement about TADs, right? Option A is they are topologically associated domains. B is they are present in interphase chromosomes. C is their formation is dependent on the SMC complexes. Or D, that is all of the above. So another thing that you have already read about, I'll give you a brief moment to ponder upon the question, and then we'll discuss the answer. Okay. So the answer for this particular question is all of the above, right? The full form of TADS is in fact topologically associated domains, right? These are both characteristics of these topologically associated domains that they are present in the interface chromosomes, right? So when these chromosomes are not undergoing mitosis or meiosis or any other kind of cell division, that is when these particular topologically associated domains are actually actively seen. Also, their formation is dependent on the SMC complexes, right? Structural maintenance of chromosome complex proteins are a key for the formation of these particular proteins, which is known as TADs, right? So, coming to question number 5, uh, the question is the histone code hypothesis essentially states that A, histone modification is important for the regulation of the cellular functions. B, each combination of histone modifications would attract specific proteins that would then execute the appropriate function. C, specific modifications on the histone tails provides stability to the nucleosome complex, or D, that is none of the above. Okay, so for this, you should be aware about what are histones, what is histone code, right? Uh, and then you'll understand what it does. So the correct answer for this one is that the histone code hypothesis essentially states that each combination of histone modifications would attract specific proteins that would then execute the appropriate function, right? So over here, I have mentioned the definition of the histone code that I have picked from this particular book by Shahid et al. Right. Also, in the second half of this lecture, I will discuss in detail about this histone code, right? So let us go through the statement. The histone code is a hypothesis which states that DNA transcription is largely regulated by post-translational modifications to these histone proteins, right? So post-translational modifications will be what? Will be modifications not to the sequence of, the sequence of protein or the sequence of amino acids. It will rather be modifications such as acetylation, methylation, etc. Right? So through these mechanisms, a person's phenotype can change without changing their underlying genetic makeup, controlling gene expression, right? So that is why a lot of times what happens is that there might be some conditions or some phenomena generally in nature wherein maybe you and I have the exact same genetic level makeup of a particular protein, but the protein function is very different in your case and in my case, right? So why could that have happened? Maybe because there are different kind of methylation patterns that are present in your protein and in my protein, right? So these are also referred to as epigenetic changes, right? So they are very, very kind of synonymous to this histone code, right? So you can read more about these epigenetic changes. Briefly, I will discuss about that also, right? Okay. Okay, this is another representation of the same. So over here, let us go through the statement again so that I can explain that illustration to you. So what was the correct option that we chose? That what is histone code hypothesis? It basically says that each combination of histone modifications would attract specific proteins that would then execute the appropriate function, right? So over here, there are some examples that have been given, right? So this is a very, very old paper. It is a 24-year-old paper, but it was one of the pioneer papers in Nature, right? So you can go and read this paper, but you should always, have an open mind when you are discussing science with anyone. So something that might have been true or might have been the norm back in 2000 might not still be true, right? There might have been changes that have occurred in it. But as an example, I have kept this illustration, right? Just to make you understand what that statement means, right? A lot of it might still be true, a lot of it might not be true anymore, right? So that is why always check your facts, always check the facts even that are being delivered. So right now, say in 2024, I am giving a lecture, and the facts that I am stating might not be true when we revisit them in, say, another 5 years, right? So there might have been more discoveries or more innovations that have come up, that have discovered newer things within ourselves, right? Due to which our understanding has changed again. So have an open mind when we discuss science, okay? So over here is an example of H3, that is histone 3, and H4, histone 4, proteins. And in this, there are some flags that are given, right? So these flags are representing acetylation, right? These circles are representing phosphorylation, and these hexagons are representing methylation, right? There are some modifications that are coexisting also. So there are at different residues. So at the same residue, it will not coexist, but at different residues, it is coexisting. For example, there are some that just have this acetylation in the 14th residue. Some have acetylation in the 9th residue. Some have phosphorylation in the 28th residue. Some have phosphorylation as well as acetylation in the adjacent, in the adjacent residues, and so on, right? So what does it do? It gives a particular identity to these proteins, right? So these are kind of patterns which will mark these proteins as to which associated protein will come and recognize them or bind to them, right? So for some of them at that time, it was known that which is exactly the associated protein that will come and bind to a protein showing this kind of a phosphorylation pattern or this kind of acetylation pattern, right? And what will be the consequent function that this protein will be performing, right? So, for example, if histone 3 is acetylated at residue 14, it will recruit a protein or a module that is bromodomain, which is essential for its transcription, right? So, any residue that has this particular kind of histone modification will undergo transcription, right? And there are different kinds of functions that are associated with different kinds of histone modifications. So I hope that I was at least able to convey this properly, right? Okay. So coming to question number 6, the question is, each nucleosome core particle consists of which one of these? A is 2 molecules of each H2A and H2B. B is double-stranded DNA of 147 nucleotide pairs long. C is 2 molecules of each H3 and H4. Or D, that is 2 molecules of each H2A, H2B, H3 and H4 along with 147 nucleotide pairs of double-stranded DNA. So this is something that was discussed in this week's lectures, as well as people who did attend lecture 4, like practice session 4, that was attended by me, that I had discussed about these histones earlier also. So you also might recall it from that particular session, right? Okay. So since it is a pretty straightforward question, let us come to the answer. So the answer is the option D, that is each nucleosome core particle, what will be its constituent? So it will have 2 molecules each of H2A and H2B, which will form one tetramer, right? It will have 2 other molecules of H3 and H4, which among themselves will form a tetramer. Like you can see over here that H2A, H2B are forming this tetramer, H3 and H4 are forming this tetramer, right? So there are 2 molecules each of these 4 histone proteins, right? Along with this, there will be a 147 nucleotide pair DNA double helix, right? And also these 2 tetramers will together combine and interact, and it will form one core histone octamer, right, around which this particular 147 nucleotide pair DNA double helix will go and bind, right? Okay. So this is a full view of this particular excerpt that I had shown you earlier, right? This we had discussed in lecture 4, in session 4. I'm briefly going to go through it again. So when we saw this beads on string structure, right? Beads on strings are what? They are nucleosomes. Nucleosomes meaning histone protein, octameric histone proteins with DNA wrapped around them and linker DNA that is connecting these two histone proteins, nucleosome complexes, right? So the entire thing from one nucleosome complex to the next nucleosome complex will be around 200 nucleotide pairs of DNA, right? If there were experiments that were done to understand what is exactly the composition of this nucleosome complex or nucleosome core particle, right? So for that, linker DNA was digested using nuclease. So it digests linker DNA and it gets cleaved and separated and digested, right? After that, what is left is this core particle with the DNA that is wrapped around it. When there is a dissociation of this core particle with this wrapped DNA with high concentration of salt, what we obtain is the constituents, that is 2 units each of H2A, H2B, H3, and H4 that form a heterodimeric. Um, uh, they are heterodimers among one another, forming forming two tetrameric complexes that interact with one another to form a octameric histone core. And along with this, there was a 147 nucleotide pair DNA double helix, right? Okay, so moving to question number 7. It is phosphorylation of histone tails occurs at which amino acid residue, right? So this is a slightly tricky question. Uh, if you have not already come across this particular, kind of histone modification question. So the options that are given are lysine, arginine, serine, or glycine, right? So which of the amino acid residues phosphorylation at the histone tail will, which of these amino acids in the histone tail can undergo phosphorylation, right? So it is one kind of histone mark. Okay. So coming to the answer is serine, right? So from this question, the first thing that might be apparent to you is that not all kinds of amino acids can undergo all kinds of histone modifications, right? So there will be some kinds of amino acid residues that can just undergo methylation. There can be some that can just undergo acetylation, some that can just undergo maybe sumoylation or phosphorylation, right? So over here, the amino acid residue, which generally always undergoes or can undergo phosphorylation at histone tails, is serine, right? So this is an excerpt which talks about some prominent types of covalent amino acid side chain modifications that are found on nucleosomal histones. Right, we will discuss them at length later, but over here you can see that if there is a serine residue, right? So a serine residue can easily undergo phosphorylation and convert into a phosphorylserine, right? So this will add a phosphate tag onto the nucleosome, that is onto the histone, that is that histone tail that it is present in, right? I hope there is not too much of background noise, right? I hope that my voice is dominating that. um okay, so moving on to question number 8. Question number 8 says, what are the main experimental outcomes of the double tethered DNA assay, right? So option A is to understand the kinetics of DNA binding and modifying protein or protein complex. B is to understand the loop extrusion properties of the modifying protein or protein complex. C is to understand the directional properties of loop extrusion by the modifying complex. Or D, that is all of the above. So for this, you will have to recall what a double theta DNA assay will be, right? The hint that I would like to give is that it was a part of lecture 3, I think, of week 6. Um, so let us go through the answer for this particular question, right? So the answer is all of the above. So what does a double theta DNA assay mean, right? So if you remember, there was this lecture in which you were being explained, how the loop formation occurs and how the loop formation usually occurs through one extrusion of a protein from one side, right? And not from both the side. Extrusion of the chromatin from one side and not from both the sides, and etc. Right? During that, there were some techniques that were talked about. One of them was the double theta DNA assay. So, what does double theta DNA assay means? Means it is tethered at two ends, right? So, if we have a DNA, we are basically fixing it at two ends, right? And then we are performing an assay wherein we are in that case, they were flowing some kind of a buffer due to which there was a directional, energy flow that was occurring. And based on that, we were seeing that DNA, how DNA is responding to that. And it was basically forming a loop kind of a structure, right? So, what are the main experimental outcomes of these kinds of assays? That is double tethered DNA assay. One, it is to understand the kinetics of DNA binding and modifying protein or protein complex, right? Second, to understand the loop extrusion properties of the modifying protein or protein complex. So over here, in that case, the protein complex that was being used was a cohesin complex, right? Condensin cohesin something, right? You can go and check that and see that it is also going to understand the directional properties of loop extrusion by modifying complex. So if you remember, this cohesin complex was used, right? That was purified and then it was used and then it was seen that, yeah, it will go and bind to the DNA, it will give a bright fluorescence. And then when the buffer flow actually occurs, we will see that a loop kind of thing is extruding, right? And when the further analysis was done, it was seen that one arm of the this complex was shortened, the DNA was shortening so that the loop can actually be formed, right? The other arm was not getting affected. So there is some kind of directional property to that also, right? So the answer thus is all of the above. So, coming to question number 9, which talks about another kind of technique, which is micro-C experiment. So, the question is, what is the correct chronological order of steps performed during the micro-C experiment? A, is it cross-linking nucleosomes, chromatin digestion by micrococal nuclease ligation, purification, and then paired end reads? Or cross-linking nucleosomes, ligation, purification, chromatin digestion by micrococal nuclease, paired end reads? Or cross-linking nucleosomes, chromatin digestion by micrococal nuclease, purification, and then paired end reads? Or cross-linking nucleosomes, ligation, purification, and paired end reads? So, I'm sure you might remember the representation that was shown by Professor Koshland, right? Okay, coming to the answer for this one, right? Sure. So, this is one of the representations that I have picked up from Wikipedia. And over here, the gist of this process is apparent. So the actual correct chronological order for the steps that are performed during the micro C experiment is first cross-linking the nucleosomes, right? The cross-link is actually done first. Then there is chromatin digestion by micrococal nuclease, right? So these cross links are then digested. Earlier, they used to be digested by the restriction enzymes in a slide, and this is a slightly modified version of that, in which the chromatin digestion actually takes place through micrococal nuclease, right? After which there will be ligation that will occur, right? So these digested chromatin will then be ligated, right? Which there will be purification and paired end reads will be obtained, right? So there is one other illustration for the same that I have kept, uh, wherein there is this cross-linked DNA. Then it is either restriction digestion is occurring over here. Like I told you earlier, this used to happen. Now in there is digestion through micrococal nuclease, right? Then there is ligation of these molecules, right? After that, there is reverse cross-link, or the paired end products are obtained, after which depending on what is the purpose, there is method specific processing, and then there's quantification that occurs, right? And also micro capture C or MCC is a chromatin conformation capture method for visualizing reproducible three-dimensional contacts of specified regions of the genome at the base pair resolution, right? These methods are an established family of techniques that use proximity ligation to assay the topology of the chromatin. So obviously proximity is something that they are using, right? So the crosslinks will not be formed between two very far apart, chromatin structures, right? So, yeah. Okay, I thought I hit this slide. This is another illustration of the same. I found it to be good, but it could be a little, uh, doubt causing. So I'll skip it, but I'll keep it in the lecture notes that I will provide to you so you can go through it. Okay, coming to our final question. So it is from our final lecture also that was given by Professor Koshland. So over here, what you have to do is select the correct outcome of the micro C technique by Professor Koshland, right? So in the experiments that were performed by them, right, or explained by Professor Koshland in his particular lecture video, what was the correct outcome or the outcomes that were obtained, right? One is base of the loops correlate with the occupancy of the CARs. Then second is there is only one type of bound cohesin populations that is static. And third is cohesin stably bound to CARs prior to loop formation, right? So is 1 and 2 correct, or is 1 and 3 correct, or 2 and 3 are correct, or all of the above are correct, right? It is slightly a memory-based question, but if you did understand the experiment, you will find out like what the answer is. Okay, so we'll discuss the answer. The answer is 1 and 3, right? So I will show you the slide. I have taken a snapshot of that lecture slide from Professor Koshlin's lecture, right? These two statements are given as it is in that particular lecture, right? Base of the loops correlate with the occupancy of the cars, and cohesin stably bound to the cars prior to a loop formation, right? This is only partly true. There is one type of a bound cohesin population, but this is dynamic, not static in nature, right? So that is why 1 and 3 are the only correct options over here, right? So these are the three statements, right? You can see the first and third are exactly the same. That is base of loops correlate with occupancy of the CARs, and stably bound cohesin at CARs prior to the loop formation, right? But there are two populations of chromosome bound cohesion that occur. I would like to correct my earlier statement. So, two populations of chromosome bound cohesion occurs. One is dynamic and the other is stable, right? So that brings us to the end of the previous year question part, right? So that will be it for this particular slide. And we'll jump on to the other portion for today, right? So let me just Yeah. So for people who are attending for the first time, let me give a brief introduction of how this particular session is being conducted. So in the first half of the session, what we have done is that we have gone through the previous year questions pertaining to week 6 lectures, right? Now what we'll do is that since these are very, very concise, compact lessons, there are often a few things that are not discussed in detail, right? So those extra things or the things that I feel are important for the knowledge of cell biology, but might not have been delved or dealt with in depth in ma'am's lectures is what we discuss over here, right? For today's class, since there were two major topics that we could have talked about. One was the chromosomal packaging and histones and how histone modifications occur, etc. And the second was the SMC protein complexes. So I have actually chosen the first part, the former part, and not the latter because SMC proteins, like you have been taught or you have been delivered these lectures by people working in the field, right? And you have been given knowledge that is up to date. And my knowledge on that particular subject or the books update might not have been updated to that level, but you have been given the current knowledge. So that is an area that I don't master in. And since you already have much of the current knowledge that has been delivered to you, I am not going to discuss about that. But instead, what I will discuss is the former part, that is basics of cell biology, right? About how the chromatin structure is developed, right? How this folding occurs. And like if you have attended my lectures before this, you might recall that in week 4 session, I had already discussed a few key points about nucleosomes, stones, etc. Right? So since I have already discussed that, and since that portion you have already gone through with Professor Loloria's lecture also, I have posed some extra questions pertaining to them. So we will begin this particular segment with those questions, right? Along with them, we'll be discussing the concept behind it, and then we will delve into what you might not have been, taught in detail in this week's lecture. Okay. Okay. So the first question for just a moment. Somebody who is trying to enter, I am unable to admit. Hold on, please. Okay, anyway. So the first question is, in the expression beads on strings, what do the beads represent? Right? Are they the DNA or the histones or the nucleosomes or none of the above? Please give me a moment. Yeah, so the answer for the first question is nucleosomes, of course, right? We have discussed this during our previous year questions also while we were discussing those particular questions. And this is a slide that I have picked from session 4, right? So over here you see two electron micrograph, images, right? The first image is the chromatin that is isolated directly from an interface nucleus, and as you can see, it is appearing as a thick, strand, or as a thick thread, right? So this is around 30 nanometer in diameter. So like this, it is around 30 nanometer. In the second micrograph picture, you can see that this shows length of chromatin that has been experimentally unpacked or decondensed after isolation to show the nucleosomes, right? So this is around 10 nanometer in thickness, right? And over here, you can see that it looks as if there are these beads or dots that are present on a string that is being that has been drawn, right? So it looks like there you have drawn a line, and on that you have drawn circles, right, or beads, right? So over here, these beads represent nucleosomes. Now, what are nucleosomes? They are a complex of the histone proteins and the DNA that is wrapped around it, right? And what is this string that we see in the middle? It is a linker DNA, right? So the free DNA or the linker DNA that is separating the two nucleosomes. Okay. So question number 2. So in a chromosome, the DNA to protein ratio by mass is equivalent to 1 is to 1, 2 is to 1, 1 is to 2, or 2 is to 3. Now, this is something I specified or laid, emphasis on while I was telling this point in session 4, week 4's practice session, right? So I hope at least the people who did attend that session would recall this. Okay. So the answer for this one is 1 is to 2, right? So this means that if we are considering that chromosome or chromatin is around, or a chromosome is 3x in mass. So 1x of the mass is DNA and 2x of the mass is protein, right? So protein actually, when we look at the mass, it it consists of around 2 times the mass of the chromosome than the DNA, right? And these two, why is it twice the mass of DNA? Because it has two kinds of proteins that are present. One half of this is histone proteins, and the other half is the non-histone proteins, right? Again, if we say that DNA is to histone proteins, is to non-histone proteins, ratio by mass is equivalent to, it will be 1 is to 1 is to 1, right? Because each of them contribute the same amount of mass in a chromosome, right? So, this representation or illustration that we discussed earlier in the PYQ session also is basically an illustration wherein the chromatin consists of DNA that is bound to both the histone, that is this kind of structure, and the non-histone proteins, these colored proteins, right? The mass of histone proteins present is about equal to the total mass of non-histone proteins, right? So this is what I just told you. And in total, a chromosome is about 1/3 DNA and 2/3 protein by mass, right? So I hope that if you did not already know, or if you did not recall this, now it is clear to you, right? We have discussed this at length in session 4. So question number 3 is nucleosomes repeat after an interval of about 147 nucleotide pairs, 200 nucleotide pairs, 100 nucleotide pairs, or 270 nucleotide pairs. Now this I have discussed even today, right? In the past 1 hour also I have discussed this particular question. Okay, so the answer for this one is 200 nucleotide pairs, right? So when I was showing you that histone, octamer, and 147 base pair long DNA in the nucleosome core complex, at the top of that illustration, I did read out that the nucleosomes repeat after an interval of about 200 nucleotide pairs, right? So coming to question number 4, which is, sorry, how many turns does DNA double helix make around the histone octamer, right? Is it 1 turn or 10 turns or 4.7 turns or 1.7 turns? Okay, so the answer for this one is 1.7 turns. That is, the DNA wraps 1.7 times, right, on the histone octamer or the histone proteins before it actually again forms that free DNA, linker DNA. Now, out of these 5, you have to pick the correct statement, right? So this is associated with the folding of DNA on the histone core particle, right? So the statements that are given over here are A, minor grooves rich in AATT18 dinucleotides are preferred at regions where DNA bends over histones. B is major grooves rich in AATT80 dinucleotides are preferred at regions where DNA bends over histones. C is minor grooves rich in GC dinucleotides are preferred at regions where DNA bends over the histones. Or D, that is major grooves rich in GC dinucleotides are preferred at regions where DNA bends over histones. So this again might be confusing for people who have not gone through this concept with me, right? So we are going to discuss that concept when I give you the answer. Okay. So the correct statement out of the 4 is that okay, why is my is that minor grooves that are rich in AATT or AT dinucleotides are preferred at regions where DNA bends over the histones. So as like you can visualize this, this might help you visualize that the octameric core complex is like round, right? And the DNA is a straight, although it is a double helix, but it is more or less straight, right? Now, what has to happen is that the straight structure has to bend over this octameric core complex that is, round in shape. It has to bend so that it can, turn around it and it can wrap around it, right? So DNA might not be that flexible at all its residues that it can bend over, right? So the residues which are preferred for this bending region are basically the minor groove region, which are rich in AATT or TA dinucleotides, right? So they are preferred over here, right? While minor grooves that are outside have GC rich regions in them, right? So this is how basically the DNA is able to wrap around the histone core complex, right? Because that is also a task. Now, this means that maybe after around every, 200 or so nucleotides, the pattern of dinucleotides that are prevalent at certain regions is the same, right? The sequences might be very, very different, but these patterns have to be conserved so that after a particular length of DNA, it can actually wrap around these histone core complexes, right? So this represents the bending of DNA in a nucleosome. Okay. So, which brings us to the end of the extra questions also. So they were, that was it for the extra questions. And now what we are going to discuss at length is the structural organization of the eukaryotic chromosomes, right? So as we have already talked about eukaryotic chromosomes, what is the key feature in this is that it is very, very condensed, right? So let us again briefly go through how, like, what is the level of condensation that is seen over here, right? So although every eukaryotic chromosome includes millions of individual protein molecules, right, each chromosome contains just one extremely long linear DNA molecule. So I hope that everybody is aware of this, and you resonate with the statement that although there are multiple individual protein molecules that are present in a eukaryotic chromosome, but that is just one long continuous linear DNA molecule that is folded and wrapped around so that it can be condensed into this form of a chromosome, right? So that is just one long DNA molecule uh, in text, right? The longest DNA molecules in human chromosomes, for instance, are 2.8 into 8 raised to power 10. So there is some typing error over here. So 2.8 into 10 raised to power 8 base pair, or almost 10 centimeter in length. Right, so this is what I told you earlier, that the longest DNA molecules are around 10 centimeter in length, right? If we are not wrapping it, if we are letting it be in this linear state, then it will go up to around 10 centimeter, which is around this long, right? And this long cell we don't have, right? We have cells which are in microns, and we have nucleus which is even smaller than the entire cell itself, right? And these DNA molecules are actually present within those, or packed within those nucleus itself, right? So, yeah, the structural organization of chromatin allows this vast length of DNA to be compacted into microscopic constraints of a cell nucleus, right? Yet, chromatin is organized in such a way that specific DNA sequences within the chromatin are readily available for cellular processes such as transcription, replication, repair, and recombination of DNA molecules, right? So, what does this mean? That uh, chromosome is a highly condensed structure, right? Even chromatin, which looks like a thread, that is also a very condensed structure, right? Although, or despite it being that condensed, um, it has DNA that is readily available for replication or transcription or repair to occur, right? So if, for example, a cell has to enter the cell cycle, so replication has to occur, right? So for that, even though it is in a very condensed or compact state, what will happen is that it will still find ways in which to decondense at a specific region so that that region can be replicated, then that will again be condensed, then the following region or the succeeding region will be decondensed, that will be replicated, and so on. This replication process also will happen, right? So despite it being very, very condensed and compact, it is readily available for such cellular processes, right? So that is a remarkable characteristic feature. When we talk about histones, right? When we talk about histones, there are 5 major types of histone proteins that are present. So they are H1, H2A, H2B, H3, and H4, right? All of these are rich in positively charged basic amino acids which interact with negatively charged phosphate groups in the DNA, right? So that aids the binding of these particular histones with DNA, right? So that means that since they are rich in positively charged amino acids, they will overall have a positive charge, right? This will attract DNA to come and bind or wrap around it, right? Because DNA has a negative charge because of the phosphate groups, right? Also, from this, you will recall that histones H3 and H4 fold into a tetramer in the absence of histones H2A and H2B, right? So they form one tetramer. Then these two heterodimers, that is H2A and H2B, associate with H3 and H4 tetramer as is seen over here, right? Also, the positive charges on surface of the histone octamer in the region that is interacting with DNA holds negative charged DNA against the surface of histone octamer at the center of the nucleosome. So this is what we have said, that since it is positively charged, it will be more successful in attracting and holding the DNA around it, right? Also, you might notice that out of all these histones, we had already previously also talked about H2A, H2B, H3, and H4, right? And we have talked about these again, right? Because they form the octameric histone core or octameric core complex or nucleosomic core complex, right? But H1 is another kind of histone protein, but we have not really talked about it. So, what is this H1? We will discuss in our successive slides, right? So, it is histone 1 and it is not really part of this octameric core complex, but it is only transiently associated with DNA when it is actually wrapping around the core, right? So, we'll discuss that briefly. Okay. So when we talk about the structure of the 30 nanometer fiber, so I hope you remember what this 30 nanometer fiber was, right? Uh, just for recollection, yeah. This is the 30 nanometer fiber, right? The beads on string is the 10 nanometer fiber, and this is the 30 nanometer fiber, which is a more condensed form of chromatin, right? Chromatin is generally present like this in the interface, right? So when we talk about this 30 nanometer fiber, its structure is not really known, right? So the arrangement of nucleosomes in the 30 nanometer chromatin fiber remains controversial because it is not exactly known that how is this arrangement happening, right? But there are two probable models that are being worked upon to explain the structure. And we are going to briefly look upon these models as well, right? Uh, Before this, um, let us discuss the role of H1. So these 30 nanometer fibers also include H1, the fifth major histone protein, right? So the histone protein that we said we have not talked about is a part of this 30 nanometer fiber, right? This H1 is bound to the DNA as it enters and exits the nucleosome core, but its structure in the 30 nanometer fiber is not known at the atomic resolution. Right, so this is just a representation or illustration of how it might look, but we are not really exactly aware of how it will look and where it might be, right? But we know that transiently when this chromatin is or the DNA is wrapping around this stone octameric core particle, it will actually present itself, right, at the entry and exit of this DNA. Okay. And okay, let us now discuss these two probable models that might explain the arrangement of nucleosomes in the 30 nanometer chromatin fiber, right? So we have said that we are not clear about what might cause this arrangement, but there are two probable models that we are going to look at, right? The first model is known as the solenoid model, right? So this is a solenoid model. What it says is that in one widely accepted model with considerable experimental support, nucleosomes are arranged in a helical conformation forming a solenoid with 6 or more nucleosomes per turn of the helix, right? So, for example, this is a chromatin structure that is coming, right? It has nucleosome, it has this histone H1 particle, and it has a linker DNA between that, right? So between two H1 particles and between two nucleosomes is the linker DNA. Now it says that uh, there might be a helical conformation. So helical meaning there might be this kind of conformation, right? Helix is always going. Uh, Let me just go it. So this kind of conformation, right, might be present, wherein there are around six. Where is this pointer? There are around six of these nucleosomes that are present per turn of this helix, right? So this is one of the possible models, the solenoid model, right? It is one of the models that is more believed in. But at the same time, the other model, which is the two-start helix model, is also something that is, experimentally worked on. There are a lot of research groups that are working on explaining if this could be the possible, model of explaining how the structure of 30 nanometer fiber is actually found, right? So in a second strongly supported model, the two-start helix nucleosomes alternatively stack like coins in the two strands, and those strands wind into a left-handed double helix. So in the solenoid model, we were saying that it forms a helix and it is looping over one another, right? But over here, we are saying that these nucleosomes are stacking like coins, right? And after they have stacked like coins, they are actually forming a double helix structure, right? So they are stacking like coins over here, and then these are forming a double helix structure. There's a similar helix that is coming from here also. Right, so this is the two-start helix model. The more accepted one is actually the solenoid model. But I have briefly told you what both these models are. Okay. So we don't exactly know till now like how this arrangement is occurring. So hopefully with better techniques being developed, like every year, every month, we'll soon find out. Okay. So when we talk about conservation of the chromatin structure, what does it mean? That the general structure of chromatin is remarkably similar in the cells of all the eukaryotes, right? So it is very well conserved across species, including fungi, plants, and animals, indicating that the structure of chromatin was optimized early in the evolution of the eukaryotic cell, right? So it is so conserved actually that there are very okay. So first I'll give you these examples, then I'll explain. The amino acid sequences for the four core histones, right? That is H2A, H2B, H3, and H4 are highly conserved between distantly related species, right? So distantly related meaning that if we talk about the thymus from a calf and sea urchin, in these two, the sequence of H3 histone is differing only by a single amino acid, right? Also, if we talk about a garden pea, right, which is not even from kingdom Animalia, it is from a different kingdom altogether. And we talk about the thymus of a calf, they both differ only by 4 amino acids, right? So we know that it is a highly conserved sequence, right? The sequence of the four core histone proteins is highly conserved. Yeah. And the amino acid sequence of H1, however, varies more often from organism to organism than do sequences from other major histones, right? So the ones that are forming the core complex, they have highly conserved sequences. H1, on the other hand, has a relatively less conserved sequence, right? Also, like it is believed that since this line is also written that this indicates that the structure of chromatin was optimized early in evolution of eukaryotic cells, right? It is believed that if there were major changes that occurred in the histone, proteins or in the histone amino acids, right? If there were multiple changes that occurred, it was probably excluded out, right? During evolution, it was not favored. So that is why still we have like very limited mm, changes in this histone core proteins, or the sequence in fact is very conserved, right? Okay. So when we talk about variations, right, although we are saying that there is high conservation of the sequence of the major major histone proteins, but there are minor histone variants that are present, right? And these minor histone variants perform specific functions, right? But like the folding or the level of folding is also different in different animals, right? Or even how often some particular cell will go into mitosis, etc., will be different in different organisms, right? The needs will be different. How often a DNA mutation can occur might differ between organisms, right? So for that, there have to be some changes that need to take place. These changes occur in the minor histone variants, right? So what are these minor histone variants? They are encoded by genes. These minor histone variants are encoded by genes that differ from the highly conserved major types. And they also exist particularly in vertebrates, right? Most minor histone variants differ only slightly in sequence from the major histones, right? For example, we will talk about variants of H2A, which will be named as H2AX. So it will vary maybe in one amino acid sequence from the major histone protein, that is H2A, right? But it will have some other role to play altogether, right? These slight changes in histone sequences may influence the stability of the nucleosome as well as its tendency to fold into the 30 nanometer fiber and other higher order structures, right? Okay. So, we will talk about some of the types of or some of the variations in this minor histone proteins. So one, uh, first let us talk about that H2A variant that I briefly gave you an example of. So a special form of H2A, which is designated as H2AX, is incorporated into nucleosomes in place of H2A in a small fraction of nucleosomes in all regions of the chromatin. So if we talk about the entire length of chromatin, right, in some of the nucleosomes, randomly or maybe not so randomly, we don't understand that very well right now, right? There will be an incorporation of H2AX variant instead of H2A, right? At sites of double-stranded breaks in the chromosomal DNA, H2AX becomes phosphorylated and participates in the chromosome repair process, probably by functioning as a binding site for repair proteins, right? So how does this variation help? That what happens, maybe it is dispersed at equal lengths, we don't know that yet, right? So wherever this H2AX is present, if at all there occurs some kind of double stranded breaks in or around that region, what this H2AX will do, it will become phosphorylated, right? And once it becomes phosphorylated, it can recruit maybe proteins or molecules that can come and sit on it and read this, and they will basically instruct chromosomal repair to occur, right, at these sites or around these sites. Uh, so that is how this variant particularly helps. Then also in nucleosomes at centromeres, right? So centromeres is the part where sister chromatids are attached to one another. So at the centromeres, H3 is replaced by another variant that is called CENPA, right? So you might come across this while reading some histone-related papers also. What does this variant do? It participates in the binding of the spindle microtubules during mitosis, right? So we have been studying about mitosis, maybe in some of the illustration that I did show to you, but I did not explain what the CENP does. What does it do? It is a variant of H3, right? What will it do? It will help in the binding of spindle microtubules during mitosis, where at the centromeric regions, right? Okay. The third instance is a variant of histone H3, which is known as H3.3, right? It replaces the major histone H3 in the transcribed regions of the DNA. Probably when the histone octaper must be moved out of the way by histone chaperones as RNA polymerase transcribes DNA in chromatin. Right. So how might this be important? Because it might help in the transcription machinery so that uh, this histone core particle knows that when H3.3 comes instead of H3, what has to happen is that the histone octamer has to be removed or it has to be shifted so that this particular region of DNA is available for transcription, right? Uh, okay. Then modifications of histone tails control chromatin condensation and function, right? So we have talked about some of these modifications earlier, right? What could these modifications be? They could be modifications such as phosphorylation in the serine residues, acetylation, methylation, and lysine residues, etc. Right? So we'll talk about them a little bit more. So each of the histone proteins making up the nucleosome core contains an intrinsically disordered, flexible N-terminus of 19 to 39 residues, which extend from the globular structure of the nucleosome. And the H2A and H2B also contain a flexible C-terminus extending from the globular histone octamer core. These termini together are known as histone tails, right? So what are these histone tails? They are basically regions that are extending out from this protein complex, right? This is what it looks like, right? So this is the core of this histone octamer, right? Around that you are seeing that DNA is bound. And these are the tails that are coming or originating in these histone octamer itself. But as you can see, they are reaching as far out as beyond the DNA that is bound around this particular octamer, right? So here is an illustration of the same. So there is an histone core complex, then there is the DNA that is wrapped around it, and the tails that are arising from, or these residues that are rising from these particular histones, they are stretching far beyond the reaches of DNA itself, right? So this might have a purpose, right? So we are going to understand what the purpose of this particular histone tails is, right? So the histone tails are actually required for chromatin to condense from the beads on a string conformation into a 30 nanometer fiber, right? So that is one of the functions of these tails, so that it can signal, or it can signal maybe proteins to come and bind to it, which will attract other proteins to bind to it, such that it will form a different kind of conformation, so that from this beads on string, it can go and condense into a 30 nanometer fiber, right? The thicker one, the chromatin fiber that is generally present in interface. So there were experiments done that indicate the N-terminal tail of histone 4. The histone 4 N-terminal tail, particularly at lysine 16, are critical for forming the 30 nanometer fiber, right? So these experiments were done in early 2000s, right? This is an example in which you don't have to look at the histone modifications right now, right? There's a key that is showing these modifications such as methylation, phosphorylation, acetylation, etc. So as you can see, in different residues, there are so many, amino acids present on their tails, right? Among these, you can see that K, that is lysine, and R, that is arginine, are pretty dominant, right? But lysine is the one that is most dominant, right, over here. So as you can see, there are a lot of these lysines that are present. And in H4, it is seen that at 16th position, the lysine that is present is actually key for the modification that will be done on this particular lysine will determine its arrangement in the 30 nanometer fiber, right? Then we talk about modifications of histone tails that control chromatin condensation and function, right? So histone tails are required for chromatin to condense from a beads on string conformation into 30 nanometer fiber. We read about this. Also, we read that lysine 16 particularly is important for, the histone modifications that will be done on it, and it is critical for forming the 30 nanometer fiber, right? Also, this positively charged lysine interacts with a negative patch at H2A and H2B interface of the next nucleosome in the stacked nucleosome of the 30 nanometer fiber, right? So what will happen? The positive charge in this lysine will probably interact with the negative groups that are present in negative amino acid groups that are present in H2A and H2B, and that will force them to come together, right? So that will help in the compaction of these structures, or maybe that will help in the loop formation also, right? Because these two beads have come together and the DNA that is left is forming a loop, right? So that is how these structures, form. Also, there are post-translation modifications on the histone tails that are present, right? We talked about this. The histone tails are subject to multiple post-translational modifications. Some of these are acetylation, methylation, phosphorylation, and ubiquitination. These are the most common ones, right? So over here, AC represents acetylation, P represents phosphorylation, UB represents ubiquitination, and ME represents methylation, right? So these are going to be post-translation modifications. What does that mean? That what is translation? Translation is the process in which a protein is formed based on the from the mRNA, based on the sequence that is present in the mRNA, right? So that will determine which amino acids will be joined to which other amino acid, and which protein will actually be formed, right? This process is known as translation. So the sequence of amino acids is actually going to form a protein. But what are the post-translation modifications that will occur, right? Most of these modifications do not alter the sequence of these proteins, right? What they do instead is that they will add tags such as acetylation or phosphorylation of certain residues. Like they will go and they will phosphorylate certain residues, they will ubiquitinate certain other residues, due to which the properties of these residues will change, right? So the sequence will remain the same, but the property or its ability to perform one function will change, right? So that is what is post-translational modification, right? For example, this is what we were talking about when we were discussing the previous year questions also, and I said I will talk about it in detail later. If I talk about lysine, for instance, right? So lysine can undergo either acetylation or methylation, right? So it will depend on the status of lysine, that whether it has undergone acetylation or it has undergone monomethylation or dimethylation or trimethylation, that what is the kind of tag that is put on this particular lysine group on the histone tail, and that will probably determine the structure that it is going to form, right? Similarly, we talked about other amino acids that might undergo different kinds of such modifications, right? So, for example, serine might not be able to undergo methylation or acetylation, right? But what is one of the common changes that occurs in serine? It can undergo phosphorylation, right? And then it can form phosphoserine, right? So a phosphate tag could easily be added to a serine group, right? Under the correct conditions. So what are the changes that these kinds of tags will bring about? So you can see that there are three different levels of lysine methylation: monomethyl lysine, dimethyl lysine, and trimethyl lysine, right? Each can be recognized by a different binding protein, and thus each have a different significance for the cell, right? So obviously they have been shown differently, right? Depending on whether it is dimethyl lysine, or maybe some protein comes and binds and it says that okay, transcription has to occur over here. So now this particular chromatin has to be opened up, right? Because transcription machinery has to come and sit on it, and replication forks have to be formed, and replication needs to be, or transcription needs to be done, right? Or maybe if it is a trimethyl lysine, what will it indicate? It might indicate that, okay, now we can form a heterochromatin structure, right? So it should be a tightly compacted chromatin structure, because this is the region where there will be no replication or transcription that will be occurring, right? It is the dark colored region, if you might remember, right? So it will have a different significance for the cell, different kinds of methylation also on the same lysine residue, right? Also, we should note that acetylation, if the lysine is acetylated, it will remove the plus charge on the lysine, right? So lysine is a positively charged amino acid, right? If it is methylated, it is still, retaining its positive charge. But if it is acetylated, it will lose its positive charge, right? And it is most importantly, uh, and it is important to note that acetylated lysine cannot then be methylated, right? It cannot have the same lysine residue will not be acetylated and methylated at the same time, right? It might at some point be acetylated, at some other point be methylated, etc. But it cannot have these two different, groups together, right? Serine phosphorylation adds a negative charge to the histone. So if phosphorylation will occur, it will add a negative charge to the histone, right? If a negative charge is added, probably it will loosen uh, uh, the DNA that is bound to it, right? DNA might not be tightly bound to this particular histone protein. With a lot of phosphorylation, it might loosen this. So maybe when transcription or replication has to occur, maybe phosphorylation, a lot of phosphorylation might be seen, right? Also, there are some histone modifications that are not shown over here. Some of them include mono or dimethylation of an arginine, the phosphorylation of a threonine, addition of ADP ribose to a glutamic acid, and the addition of a ubiquityl sumoyl or biotin group to a lysine, right? So these are not very frequently occurring. They also occur, right? And there will be sufficient number of modifications in these residues and these modifications in these residues, but they are not as abundant as the ones that we have just discussed, right? And then we come to this histone code, right? So we have briefly talked about this also. What is a histone code? It is basically a signature of these post-translational modifications on the histone, right? Let us read as to what it is. The particular combinations of post-transcriptional modifications found in different regions of the chromatin constitute a histone code that influences chromatin function by creating or removing binding sites for the chromatin-associated proteins, depending on the specific combination of these modifications that are present, right? The histone code of modified amino acids in the histone tails is read by proteins that bind to the modified tails and in turn promote condensation or decondensation of chromatin, right? There could be other kinds of things also that is occurring. If you remember from our PYQ session, I showed you that one table from a Nature paper back in 2000, right? There it was written that, okay, if in H3, if there is a acetylation on residue number 8, it will probably recruit this particular domain and that will probably cause transcription, right? So, you can pick up these examples from older, newer, maybe review articles, papers, books, etc. Right? Okay. When we talk about euchromatin and heterochromatin, I hope you remember heterochromatin is this densely colored region wherein the chromosome is also very, very, or chromatin is very, very densely packed. And since it is so densely packed, it is not really available for transcription or replication to occur. So these are transcriptionally repressed sites, right? Euchromatin, on the other hand, is the region where these chromatin is loosely bound on the histones, and thus it is a transcriptionally active region, right? So over here, this phenomena is also explained with this histone code or histone modification. For example, they are saying that if in histone 3 at the ninth residue the lysine is methylated, right, it will probably be responsible for heterochromatin to be formed, which will be a transcriptionally repressed or inactive or condensed region, right? Similar is the case for methylation of lysine at residue 27 of histone 3, right? On the other hand, right, if acetylation is dominating this methylation, right? So in the same histone protein, you can see there is the same sequence that is given over here. If at residue 9, instead of methylation, there is acetylation, right? Along with that, there are some more acetylation tags that are present, and there is phosphorylation and less of methylation, right? Then it will probably be a more open kind of structure, and it will be transcriptionally active, right? And this will form the euchromatin. Also, this will prove what I was trying to tell you earlier, that at the same region, at the same time, it cannot be methylated and acetylated, right? It can either be methylated or acetylated. Even at different points, it could happen that first it was methylated and it was in a heterochromatin region, and then there were some changes that occurred and it converted back into the methyl group was removed, then there was just lysine that was present, then acetylation happened, then acetyl group was added, and on after that this became a loosely packed structure due to which it became transcriptionally active, etc., right? So this is a very dynamic process, it can keep changing, right? But mostly the regions that are heterochromatic are kind of conserved, right? uh, which and why they are conserved? They are conserved because of the memory, which is also known as epigenetic memory. Now, why is it known as epigenetic memory? Because these modifications, like I told you, is not happening in the sequence of the amino acids, right? It is happening on the tags that are present on the amino acids, right? These tags are not genetic in nature, right? They are very, very environmental or circumstantial in nature, right? So they are epigenetic factors that are affecting this particular chromatin. As to whether it will remain heterochromatin or euchromatin, whether it will be transcriptionally repressed or transcriptionally active, etc., right? Please give me a moment. So when we talk about epigenetic memory, let I give you a brief introduction. Now let us read what is written over here. Right. So this has been picked up from Lodish. The model of heterochromatin formation provides an explanation for how heterochromatic regions of the chromosome are re-established following DNA replication during S phase of the cell cycle. So what does this mean? That if you are not already aware, after DNA will be replicated. So whenever it is being replicated, obviously the entire length of DNA has to be replicated, right? But whenever it is transcribing, right, the entire length of DNA will not be responsible for transcribing anything into an mRNA, right? There will be short regions of the D or that are present on the DNA, right? These will be the genes, right, which will transcribe something, and there might be multiple of the genes that are present on a single DNA molecule, right? And not the entire DNA again will be transcriptionally active, right? So after this replication of the entire DNA is done, whatever was a transcriptionally inactivated region, again it will form this very, very dense structure, and again it will become a transcriptionally inactivated region. Now, how does this region know that, okay, I don't have to be transcriptionally active, right? So that is because of what we call as epigenetic memory, right? When DNA in heterochromatin is replicated, the histone octamers that are di or trimethylated at H3 lysine 9 become distributed both the daughter chromosomes along with an equal number of newly assembled histone octamers, right? So we saw that it is one of the characteristic marks of heterochromatin that at lysine 9 or at lysine 27, these particular, you know, residues, lysine residues will be methylated, right? So they are di or trimethylated at that also. And when replication is occurring, they will be equally distributed among uh, no, among the parent and the daughter cell, right? Then the H3K9 HMT associated with the H3K9 dimethyl or and trimethylated nucleosomes will methylate lysine 9 of the newly assembled nucleosomes and then regenerate the heterochromatin in both the daughter chromosomes, right? This will consequently the chromatin will be marked with an epigenetic code, so called because it does not depend on the sequence of the bases in DNA that maintains the repression of associated genes in replicated daughter cells, right? So I will give you an illustration of what is given, right? So when replication is occurring, what will happen is that this histone H3K9 methyl transferase will transfer some methyl groups to the newly replicated region, right? This is the heterochromatin region that we are talking about. Then there will be binding of this HP1 chromodomain to this H3K9 trimethylated groups, right? And once this happens, this HP1 and trimethyl, this complex, it has a tendency to go and to attach to or interact with other HP1 trimethyl complexes, right? So because of which it will form it will cause oligomerization of HP1, right? Once it will go and interact with so many domains, what it will do is that these domains, even if they are present far apart in the chromatin, it will be brought together, right, into a more condensed form. And that is what heterochromatin is, right? On the other hand, if acetylation is present instead of this HP1 and trimethyl tag, what will happen is that it will act as a euchromatin, right? It will be a more loosely bound chromatin structure, right? So that is how this particular phenomena takes place. I wanted to talk about lamp brush chromosomes also, but I have not really prepared that slide. So I am just giving you an introduction of lamp brush chromosomes. So these are the chromosomes in which the chromatin looping has most often or most commonly been or most distinctly been observed, right? So in a electron micrograph, for example, you can see that if we see the lamp brush chromosome at the interface in the nucleus of an oocyte from a newt, Notophthalmus viridens, then we can see that there are very, very significant coilings of these chromosomes that occur, right? So this was one of the first, representations wherein non-histone proteins were shown to organize long chromatin loops, right? After this, similar phenomena has been studied on vertebrates, mammals, etc. And it has been seen according to the SMC proteins involvement that you have seen in this week's lecture, that such a phenomena may also occur in case of higher order organisms, you know. Okay, so like I said, I wanted to emphasize a little more on Lambrus chromosomes, but I won't do that without concrete statements, because I don't want to keep you guys hanging or deliver some incorrect information. So we'll begin with this part in our next lecture, right, in session 7 after your week 7 lectures. So that is it from my end for today. I hope I was able to deliver like what I meant to uh, in this extra concept part specifically, right? and I hope that that did add to some of your knowledge. Uh, oh, I'll stop sharing my screen. Yeah. Okay. So having said that, uh, that brings me to an end of this particular lecture. I do hope that like I said earlier, I was able to deliver or convey what I meant to in this session, and I hope you guys understood it. If you have any particular queries regarding this session or regarding other sessions also, you may ask. Okay. So in that case, what I'll do is that I'll just stop the recording and I will end the session. Thank you all for being a part of the session, right?